Swayam Prabha. Digital India. Educated India.
The first step that when we discuss about the chemical origin of life or chemogeny is formation of simple organic compounds. How the simple organic compounds were formed or how they came into existence. The first step is the formation of hydrocarbons. Remember, we discussed there was an abundance of hydrogen. There were other gases like methane or ammonia. So these gave rise to simple molecules like acetylene or ethylene and so on. From these simple hydrocarbons, then various derivatives, particularly the oxy and hydroxy derivatives of these hydrocarbons came into existence, such as the acetaldehyde, ketones, various acids, etc. From these derivatives of hydrocarbons, the carbohydrates were formed. Carbohydrates, as you are aware of, they are the small chains of compounds of carbon, hydrogen and oxygen. From these carbohydrates, then fatty acids and glycerols were formed because there were the reactions of condensation, polymerization and oxidation which gave rise to long chains of carbohydrates, ultimately the fatty acids and glycerol. Now from these combination of hydrocarbons along with the ammonia and water gave rise to amino acids. So in a sequence of these 5 basic steps, you can appreciate how simple hydrocarbons were formed first, leading to derivatives of hydrocarbons, then carbohydrates, fats and amino acids, which are the simple molecules, were formed. Again, a sort of recap to what we discussed about biochemical origin of life or biogenesis particularly, which was supported by the experiments given by Stanley Miller and Harold Uray, or popularly called as Miller-Uray experiment. Let's quickly revise these experiments. Remember, if you heat the water, which resembles the primitive ocean, so you provide heat here, the water vapor circulate or move from this path, and under the effect of this electric spark, which shows the kind of lightning effect, the primitive atmosphere composed of water, methane, ammonia and hydrogen, when these molecules get condensed or get cooler, if you take the samples from here, you can see the simple molecules which we just discussed, the simple amino acid, the hydrocarbons, the fats and simple carbohydrates you can observe in this sample. So this was an experimental proof for how the simple molecules came into existence on the primitive earth. Now Haldane gave a term called hot dilute soup or the primordial soup. Now this term was primarily used for the sea basically, because all these small molecules which we discussed now, they were getting formed in those primitive aquatic bodies or sea primarily. Let me just quickly read it for you. The aqueous solution of organic compounds that accumulated in primitive water bodies of primitive earth as a result of endogenous abiotic synthesis and the extraterrestrial delivery by the comets and meteorites collision, and from which some have assumed that the first living systems evolved. So this is a term given by Haldane to see where the life originated in the form of simple molecules preliminarily. The next step in chemogeny is formation of complex organic compounds or the macromolecules from these simple molecules which we just discussed. Now, these aquatic bodies had large amount of these simple molecules. So, it is possible that collision reactions, aggregation reactions started happening in that. Because of which, the simple sugars formed di and polysaccharides, fatty acids, glycerols formed fats, amino acids formed polypeptides, etc. So it is quite possible that because of various reactions, the small molecules got converted to organic compounds or the biomolecules. Then the question comes, what came first? Was it the RNA or was it the protein that came into existence first? There are two schools of thought. The one says there is an RNA world or RNA first hypothesis, and it has been proposed by Leslie Augill, Francis Crick and the Carl Woese. According to them, it was the RNA which came into existence first. They say that RNA world is the first stage in evolution of life. It has catalyzed all molecules necessary for survival and replication. The autocatalytic RNA were named as naked genes. But the question comes, without proteins, how RNA formed? Because you might be aware of the central dogma. For RNA to form from DNA or the transcription process, you require lots of proteins. So how the RNA came into existence if there was no proteins? Another objection has been put forward is RNA is too complex to have arisen prebiotically. It is a complex molecule. So in prebiotic conditions, how it came into existence? And third, biochemically, if you see, RNA is not a stable molecule. So how it got the stability in that atmosphere? These are some of the questions. But besides this, remember, there are many supporting evidence in the form of for the RNA world hypothesis. First one is the enzymatic activity that ribozymes do have. Remember, whenever we talk about proteins, we say all enzymes are proteinaceous. So the ribozymes, which are nothing but the RNA molecules having the catalytic properties or enzymatic properties, so they also exist in nature. Second thing is, RNA in ribosomes may catalyze peptide bonds. So here RNA is performing the catalytic activity. Quite possible that in primitive earth, there also RNA formed first and it had the catalytic activities. Also, if you focus here, all the essential molecules in biochemistry or metabolism, the NAD, FAD, coenzymes, etc., they are also derived from RNA. And there are examples available where RNA acts as a gene regulator. So all these evidence support the view of RNA first or RNA world hypothesis. In contrast, there is a protein first hypothesis also, which has been given by or supported by many of the biologists, including S.W. Fox. According to this hypothesis, the protein catalytic system must have evolved before the nucleic acid based catalytic system. Again, the question comes, how did the protein came into existence? How did they form if there was no nucleic acid, if there was no DNA or RNA? How the protein formed? Where it got translated from? But again, there is an supporting evidence for the protein first hypothesis as well, which people have cited. There is something called non-ribosomal protein synthesis. For example, in the bacteria Bacillus brevis, the antimicrobials, tyrosidins and gramicidins are synthesized in the absence of mRNA. So, there is a non-ribosomal protein synthesis as well, which does not require RNA molecule to form the proteins. So, irrespective of what actually happened, these two schools of thought we need to discuss when we are talking about chemogeny. Let's now move to third part of our discussion in the chemogeny. After simple molecule formation and the large or macromolecule formation, which is the formation of protocells. Remember, the macromolecules themselves cannot have the property of a cell. For a property of cell, we need to have some membranous structure, membranous droplets. Means from the molecules, now we are moving to droplet level of organization. Based on the structure, the surrounding membrane can exhibit selective permeability, which is very much important for existence of cell and further the life. The cell needs to have that selective permeability for survival. A self-assembled membrane-bound system containing molecules is something which we term as protocells. And each protocell, remember, must have attained the living attributes. What are these living attributes? The protocell must be able to maintain its individuality. Now when I say individuality, I am referring to selective permeability. It should be able to decide which molecules to take in and which molecule to discard off, which molecule to be allowed or not allowed to enter the cell. Also, these should be able to grow in size. They should be able to divide as well. If a cell or not cell, if a system has these kind of living attributes, then it can be called as protocells. That means it can give rise to the cell. There are two possibilities which can be discussed for the formation of these protocells. These fairly simple, artificial and laboratory based protocell type structures were devised by Operin, which he named as coeservates, and another system by Fox, which he named as microspheres. So Operin's coeservates and Fox microspheres are basically laboratory based artificial protocell system, but they are a sort of evidence that these kind of structures could have existed on Earth for the origin of life. Let us first talk about the coeservates. Coeservates are a spherical aggregation of colloidal particles in liquid suspension. They form in response to spatial conditions of, it might be the acidity, it might be the temperature, the salinity, the osmotic concentration, etc., etc. And they are dispersed colloidal particles separating spontaneously out of solution in the droplet forms. Remember, we talked about droplet kind of organization. In the presence of more than one type of macromolecule in colloid, complex coeservates form and possess various properties as well. Now it will be more clear when we discuss the basic properties of coeservates. They are simple but having a persistent organization. They remain in solution for extended periods and they do have a property to increase in size. Besides this, Operin synthesized coeservates that could incorporate various enzymes in them and they could further perform various functions, catalytic functions. Observe this diagram. Now consider this as a coeservate. A colloidal particle is there, and as I said, there can be special conditions when it can form. So here the condition is the concentration of solute. This selected molecules are getting in and the water molecules are getting out, and this is sort of a membrane. When there is a high concentration of selected molecule, so you can see this red dot which represent here the solute molecule, they are getting inside. The water molecule is more of outside. Now here you can see there are a number of colloidal particles inside because these colloidal particles can interact with each other. For example, if one is histone and another one is DNA, they can interact with each other, they can get attracted to each other, and they can get attached with each other, which may increase their size as well. And as I just discussed, the operin could successfully incorporate various enzymes also in these coacervates, which could perform various functions. For example, if you take a mixture of gum arabic plus histone plus the phosphorylase enzyme, the coacervate, the colloidal particle which forms, if you incorporate glucose 1-phosphate enzyme also in that, because of this phosphorylase enzyme, there is a polymerization of glucose that takes place, because of which you can see a starch accumulation here. Now in the second stage, if you take gum arabic, histone, phosphorylase and an additional enzyme here which is beta amylase, the coacervate that forms, if you incorporate glucose 1-phosphate again, obviously it forms this starch boundary, starch molecule there, but because of your amylase addition here, it hydrolyzes this starch into maltose. So why I am showing this experiment is, as I just pointed out, operin could incorporate various enzymes into the coacervate molecules, the colloidal molecules, which could perform various functions also. Now you can appreciate, if this coacervates form in the primitive life and the enzymes start evolving, they could attain the functions, catalytic functions. Moving ahead to the another type of protocells which Fox could develop, they are called as microspheres, and around 10 to the power 8 to 10 to the power 9 microspheres per gram of polymers are formed when thermally produced polymers are boiled in water and cooled. So he made proteinoid microspheres which were uniform in size, quite stable, they were bounded by double membranes, and they could undergo fission and budding. Thus, they have selective absorption properties, selective permeability was there, the growth property was there, growth in terms of size as well as mass, osmosis was there, rotation was there, movement were there. So, these are all the living attributes which indicate that these protocells could have originated the first cells. If you compare the proteinoid microspheres as developed by Fox with the coacervates as developed by operin, the basic procedure is depicted here. If you take amino acids, heat them, they form the proteinoids. With the addition of water, you can see the proteinoid microsphere formation. In case of coacervates, if you take mixed polymers, just like we take gum arabic, enzyme and histone we showed, if you add water and you agitate it, mix it, then you can see the formation of coacervates. With this much introduction about how simple molecules, then complex molecules formed, and how the protocells formed, now let's move to the next part of our discussion, which is the biological origin of life, or something which we call as biogeny. Now again we'll discuss this topic under 4 subheads. First is from these protocells where we left the chemogeny, how the methanogens, the first molecular fossils, formed. From there, the cyanobacteria or the stromatolites, the first fossil cells, how they formed. From there, how the eukaryotes came into existence from these prokaryotes. We'll discuss the endosymbiotic theory here, and lastly we will discuss from these unicellular organisms how the multicellularity could be achieved. Now before we move ahead to discuss all these 4 points, I would like you to give a sight to this timeline which has been shown here. Around 4.3 billion years ago on Earth, the volcanoes, comet impact and water formation took place. The secondary atmosphere. What is secondary atmosphere? I'll discuss in the next slide itself. But it formed at around 4.2 to 3.8 billion years ago. Methanogens, the first molecular fossils that I just named a few seconds back, it came into existence at around 3.5 billion years ago. Cyanobacteria came around 3 to 3.5 billion years ago. Then at around 2.5 billion years ago, we found the first bacterial fossils. 2 to 2.3 billion years ago, there was a progressive increase in atmospheric oxygen because of these organisms. And the first eukaryotic organisms came into existence at around 1.5 billion years ago. Now, as I just talked about the secondary atmosphere, the Earth's atmosphere has been divided into two, that is primary or the first atmosphere and the secondary atmosphere. The primary atmosphere was there at around 4.6 billion years ago. It was composed of hydrogen and helium. The low gravitational force of Earth, strong winds coming from Earth or Sun might be responsible for the loss of this atmosphere. But remember, there was no life form available at that time. So, none of the life has actually seen this primary atmosphere. In contrast, the secondary atmosphere was composed of water vapors, carbon dioxide with some hydrogen, nitrogen, ammonia, methane, etc. And it arose at around 4.2 to 3.8 billion years ago. Still, at that time, there was no or very little oxygen. But remember, the origin and evolution of various life forms kept on changing this secondary atmosphere. So this no or little oxygen sooner or later turned to oxygen rich atmosphere as well. Coming to our first organism from the protocells, that is methanogens. We call them as the fossil molecules. Remember, we are not talking about the fossil cells here, but the fossil molecules. Because no methanogen as such has been found as fossil. But these are the methane originating single-celled organism that could survive in absence of oxygen. And as I just said, methanogens are not known from fossilized cells, but from fossilized molecules. People have detected the methane at around this point of time, which can be of biotic origin. That means this methane could be of methanogens, or it can be a abiotic origin also. For example, some collision through comet and this methane was struck there, which people have identified at this particular time. So there is a possibility that methane which has been found, it could be because of the biotic origin of methanogens or the abiotic origin, and that is why we are calling it as the fossil molecule, not the fossil cells. Then before moving ahead, we first need to discuss how the metabolic pathways, how the nutritional pathways actually evolved. Simple anaerobic organisms which were dependent on energy source in the primordial soup, they were the first one to exist. From them, the autotrophs came into play. Autotrophs were capable of generating organic compounds using atmospheric carbon dioxide. From the autotrophs, photoautotrophs came into play, which generated organic compounds using light as the energy source. And then the aerobic organisms came into play, who were the energy drivers from the transfer of electrons to oxygen. That means they were using oxygen here. So after methanogens, let us move forward to the earliest fossil cells known, which are of cyanobacteria. The cyanobacteria, or the oldest cyanobacteria, have been found in unmetamorphosed rocks, which we call as scherts. They are associated with layered organic deposits, which we call as stromatolites. So these stromatolites are mats of organisms that traps various aqueous sediments and then they cement together to form a laminated giant knob-like structures. So, the earliest cyanobacteria which have been recorded so far, they were found in the form of these stromatolite structures. Moving ahead, when we are aware of these kind of cyanobacteria could have existed like this, next question comes, how the eukaryotes came into play from these prokaryotic organisms. I would like you to just recall the difference between an eukaryotic cell and the prokaryotic ones. You might be aware that it lies mainly with the organelles. So, membrane-bound organelles are there in eukaryotes which are not there in prokaryotes, besides other differences like cell walls, flagella, etc. etc. So, the question comes, how these eukaryotes originated from the prokaryotes. Additionally, we can refer it as how the organelles evolved in the eukaryotic organisms. And the answer lies from endosymbiotic theory, which is also called as symbiogenesis. And this theory was given by Lynn Margulis in 1967. The ancestral anaerobic eukaryotic cells. Remember, we are talking about eukaryotic cells, but anaerobic and the ancestral ones acquired the ability to engulf or endocytose and ingest aerobic prokaryotic cells. So here we are talking about the where the eukaryotic cells could engulf the prokaryotic cells, and these prokaryotic cells were aerobic in nature, whereas the eukaryotic cells was anaerobic. With time, both of these cells acquired symbiotic relationships. The endosymbiont aerobic prokaryotes, which was engulfed by these eukaryotic cells, gave rise to mitochondria and chloroplast. Now, this theory of Lynn Margulis can be understood by this simple diagram. If you see here, this is the proto-eukaryotes, and as you can see, there is no membrane-bound structures, there are no much organelles here. But in a symbiotic event, focus here, there is a symbiotic event where the aerobic bacterium is being engulfed by this eukaryotic cell. And then this photosynthetic bacteria, once it is ingested, the modern photosynthetic eukaryote was formed. Whereas if it was not a photosynthetic bacteria, it remained as mitochondria in case of modern heterotrophic eukaryotes or the animal cells. So you can see this line depicts your photosynthetic cell, eukaryotic cell origin or the plant cell origin, and here you can see the heterotrophic eukaryote or the eukaryotic animal cells is being formed. Now, once the eukaryotes also formed, remember, still they are single-celled organisms. But now what we see is the multicellular organisms. And this multicellularity has various advantages. Over the single-cell organisms, they are much strong. The strength-wise, they are most favorable. Also, they do get the protections. So, how this multicellularity got achieved in this single-cells eukaryote? The origin of this multicellularity would have required the single cells that divided into two cells to develop mechanisms. So just imagine a cell has divided into two. So there are two possibilities that it lead to eukaryotic multicellularity. The first possibility is that after division, there is a prevention of separation of the two cells. So the two cells of the same genetic material, they remain attached, and after division, they remain attached with each other. So they can give rise to multicellularity. Another possibility is facilitation of cell-to-cell aggregation. So that means number of single cells do aggregate with each other to form the multicellular organism. But in this case, unlike the previous one, the different genetic composition cells do get attached with each other and give rise to multicellular organisms. So these are two possibilities which we can discuss that could have led to the formation of multicellular organisms. So let us move to summary of this lecture. We started with theories of origin of life, where we discussed the preliminary theories of spatial creation, panspermia, biogenesis, and then we started with biochemical origin of life, which we divided into different parts. The first part dealt with origin and evolution of universe by Big Bang theory or Big Bang hypothesis. Then we moved on to discuss origin of solar system and planets including our Earth using nebular hypothesis, under which we discussed collision theory and condensation theories. After that, we discussed the chemogeny part of biochemical origin of life, where we discussed how the simple molecules, simple organic compounds form, leading to formation of complex compounds, and then the formation of protocells in the form of coacervates or microspheres. Lastly, we discussed the biogeny part, where we talked about methanogens, the first molecular fossils, cyanobacteria as the earliest fossil cells in the form of stromatolites. Then we discussed how endosymbiotic theory explains origin of eukaryotes from the simple unicellular prokaryotic organisms, and lastly how the multicellularity could have achieved from these prokaryotic single cell animals. Now these are some of the readings which you can have in order to enhance your knowledge. Particularly this website I would like to suggest if you want to have more knowledge about origin of universe. Otherwise, these books by Ridley, Hall and Hallgrimsson and the Fukuyama gives a good overview of all these biochemical origin of life. Thank you and happy learning.